Go.
With the recent advance in the DNA sequencing technologies, it has initiated an era of personal genomics. 8 human genome sequences were reported from 3 distinct geographical regions, 1 African, 4 European, a Han Chinese, and 2 Koreans. The dataset will expand as the 1000 genome project is completed. Evolutionary perspectives believe that the modern genomics is restricted from not being able to uncover past human genetic diversity and composition directly. Being able to access this data, ancient genomic sequencing is needed. The closest to the ancient human genome is 2 data sets of DNA from a single Neanderthal. 2 things that could compromise the possibility of a high sequence depth or contamination and DNA degradation. No ancient nuclear genome has been sequenced deeper than 0.7 times. This level is insufficient for genotyping and excluding errors in the sequencing or postmortem DNA damage. In 2008, a permafrost preserved hair from an early individual that settled in the New World Arctic was used to generate the first complete ancient human mitochondrial DNA genome. 80% of the recovered DNA was human and showed no sign of modern human contaminant DNA. This specimen is an excellent candidate for which the sequence of the first ancient human nuclear genome is developed. The specimen was used for genomic sequencing, and it is the largest of 4 human hair tufts excavated directly from culturally deposited permafrost sediments. Even though the hair tuft is old, using light and scanning electron microscopes, it indicated an excellent overall preservation. Miscoding liaisons make it difficult to tell which is true evolutionary derived substitutions from those that are damaged based, especially if the sequence depth is low. The results from this experiment will allow an estimate of the overall damage rate of the genome with less than 1%. It was found that the undamaged sequences to be slightly shorter on average than those containing damage. The ancient human DNA is particularly prone to contamination by modern DNA. The results confirm that the DNA preservation in the hair is an excellent judge of the ancient DNA standards, as well as using a decontamination protocol. Indexing adapters and primers were used to identify if any of the samples were contaminated after leaving the DNA Clean laboratory in Copenhagen. This would
Go.
Several indexed enrichments were carried out. This resulted in a yield of 3.5 billion reads. If any sequence did not obtain a 100% match in the indexed read, they were excluded from the analysis. The result allowed 93.17% of the reads to be attempted as mapping of the human reference genome. The primer trimming was carried out as an integrated part of the mapping during the alignment of each read to the genome. The read with the multiple matches or no matches were discounted. The reads with no matches indicated were unidentifiable, whereas the rest were of microbial, eukaryote, viral or bacterial origin. The genomes with the same start and end positions were considered colonial. This resulted in the final dataset of 28.4% of all reads. The origin of the individual, other cultures, and the relationship to present-day populations has been debated since they were first discovered in the 1950s. The genome sequencing of a 4,000-year-old human was successful. This was supported by the private SBN analysis that indicated contamination levels to be less or equal to 0.8%. This observation combined with indexing and handling indicated that contamination in the genome is not of concern. This study demonstrates that it is possible for the sequence of the genome of an ancient human to be at a level that allows the SPN and population analysis to take place. This also reveals that the genomic data can be used to identify important phenotypic traits of an individual from an extinct culture that left only minor morphological information behind. This report was performed in 2010, and there has been no further developments recorded so far.